Ground-based space environment simulation devices, a comprehensive overview. Introduction. Ground-based space environment simulation devices are essential for understanding how materials and biological systems respond to the challenging conditions encountered in space. These simulators replicate microgravity radiation and other environmental factors, allowing researchers to conduct experiments that would typically only be possible during spaceflight. This report synthesizes information from various sources to provide a comprehensive overview of the capabilities, technologies, and implications of these devices. Importance of ground-based simulation. The necessity of ground-based space environment simulation arises from the limited opportunities for conducting experiments in actual space conditions. Microgravity research has demonstrated its critical role in uncovering the effects of gravity on biological processes and material behavior. Due to the scarcity of spaceflight missions, ground-based simulators have become invaluable tools in astrobiology and materials science. Types of ground-based simulation devices. 1. Material testing simulators. Ground-based facilities like the ITL Innovative Technologies Laboratory offer a range of simulators designed to test materials under conditions that mimic low Earth orbit, LEO, lunar, and deep space environments. Some notable facilities at ITL include UltraMARS and UltraPeen. These are advanced simulators that expose materials to atomic oxygen and other space-like conditions. Fast atomic oxygen facilities. They simulate the exposure of materials to fast atomic oxygen, which is crucial for evaluating the durability of spacecraft components. Lunar dust simulators. These facilities test how materials interact with lunar dust, aiding in the development of protective technologies for future lunar missions. ITL has collaborated with major space agencies including NASA and ESA, testing over 65 different materials, providing crucial data on their performance in space. 2. Biological research simulators. The impact of microgravity on biological systems is investigated using various simulators that create conditions resembling those experienced in space. Key technologies include 2D Clinostats. These devices rotate samples to minimize the effects of gravity. They have been used extensively in plant and microbial research to study growth patterns and cellular responses to simulated microgravity. Random positioning machines, RPMs. These simulate microgravity by randomly changing the direction of gravity acting on the samples. They have proven effective for studying larger organisms including Drosophila and Arabidopsis thaliana. Rotating wall vessels, RWVs, designed for cell cultures. RWVs provide a low-shear environment that mimics microgravity. They have been instrumental in studying the behavior of various cell types under simulated conditions. Comparative effectiveness of ground-based simulators. Material testing. Ground-based simulators have shown varying effectiveness depending on the material being tested. For instance, the UltraMARS facility has been particularly successful in assessing the durability of polymers and composites under space-like conditions. Meanwhile, the lunar dust simulators help in evaluating the wear and tear on materials that will come into contact with lunar regolith. Biological research. The choice of simulator can significantly affect experimental outcomes in biological research. For example, 2D Clinostats have been shown to effectively simulate microgravity for unicellular organisms like Paramecium and Euglena, maintaining their swimming patterns akin to those observed in actual microgravity conditions. RPMs, while useful, may induce additional mechanical stresses that could confound results, particularly in studies involving rapid biological processes. Researchers have identified that the 2D clinostat is generally superior for simulating microgravity conditions for most biological organisms, while the RPM is better suited for larger specimens. Collaboration with space agencies. Extensive collaboration between research institutions and space agencies has validated the effectiveness of these simulators. For example, joint studies have led to the identification of the biological responses to microgravity in various model organisms, influencing the design of experiments for future space missions. Timeline of development. The evolution of ground-based space environment simulation devices can be traced through several key developments. Late 1800s, the introduction of the Clinostat, the first device designed to simulate microgravity. 1970s to 1980s, the development of advanced rotational devices including 3D clinostats and RPMs. 2000s present, the integration of multifunctional simulators like RWVs in high-vacuum facilities. Combined with advances in materials science and biology. Current research focus. Ongoing research continues to leverage these simulators to explore a variety of scientific questions, including the effects of microgravity on gene expression and cellular processes in plants and animals, the durability of materials in spacelike conditions, particularly for future missions to the Moon and Mars, the development of protective technologies to safeguard astronauts and equipment from the harsh space environment. Conclusion. Ground-based space environment simulation devices are pivotal in expanding our understanding of how materials and biological systems behave in space. Through diverse testing methods and collaborative efforts with major space agencies, researchers are continually enhancing our preparedness for the challenges of space exploration. The insights gained from these simulators will be instrumental in the design and execution of future space missions, ensuring the safety and success of human endeavors beyond Earth. Ground-based space environment simulation devices are essential for advancing our understanding of how biological and material systems respond to the unique conditions of outer space. Here are some counterintuitive insights regarding these devices and the research conducted with them. 1. Microgravity is not truly zero gravity. A common misconception is that microgravity environments such as those achieved in ground-based simulators replicate a zero-gravity condition. However, true weightlessness can only be experienced in space. Ground-based simulators can only create conditions where the effects of gravity are significantly reduced, but not eliminated. This means that biological systems may still experience residual gravitational effects that can influence their responses differently than they would in actual microgravity. 2. Different simulators produce varied results. It may seem logical that any simulator designed to mimic microgravity would yield similar results across different biological systems. However, research shows that various simulators like 2D clinostats, RPMs, and magnetic levitation devices can produce inconsistent results even for the same biological response. For instance, while some organisms might respond well to a 2D clinostat, they might exhibit different behaviors or physiological responses when subjected to conditions in a random positioning machine, RPM. This indicates that the effectiveness of a simulator can vary based on the specific biological system being tested. 3. Fast rotation may induce stress responses. It might be expected that faster rotation speeds in devices like clinostats would improve the quality of microgravity simulation by more effectively randomizing gravitational forces. However, increasing rotation speed can also lead to mechanical stress that biological systems may perceive as a form of stimulation, leading to stress-induced responses rather than the expected microgravity effects. This underscores the complexity of simulating microgravity and the need for careful calibration of simulator parameters. 4. Magnetic levitation has limitations. Magnetic levitation is often viewed as a cutting-edge method for simulating microgravity without the lag time associated with other methods. However, studies indicate that while magnetic levitation can effectively reduce gravitational effects for some organisms like Drosophila, it can also produce artifacts due to the strong magnetic fields involved. These magnetic effects can interfere with biological processes, complicating the interpretation of results and potentially leading to misleading conclusions about microgravity responses. 5. Biological responses are highly context-dependent. Researchers might assume that biological responses to microgravity would be uniform across different cell types or organisms. However, evidence suggests that factors such as the developmental stage, genetic background, and environmental conditions can significantly influence how an organism responds to simulated microgravity. For example, cells in suspension may react differently than adherent cells, and various strains of the same species can display divergent behaviors under what is assumed to be the same simulated conditions. 6. Stress responses can mask microgravity effects when testing biological systems in simulators. The stress induced by the simulation itself can obscure or mimic the effects that would be observed in actual microgravity. This can make it difficult to determine whether observed changes are due to microgravity or are merely responses to the mechanical or environmental stresses introduced by the simulator. The complexity of these interactions highlights the need for well-designed control experiments and careful interpretation of data. 7. Historical perspectives on simulator development. The development of simulators has often been driven by technological advancements rather than a clear understanding of their limitations or the specific needs of biological research. For example, while 2D clinostats have been in use since the late 1800s, improvements in understanding the nuanced effects of gravity on living systems have only recently led to optimized designs that better meet experimental needs. This evolution suggests that initial assumptions about simulator effectiveness may have been overly simplistic. 8. Interdisciplinary collaboration is essential. The most effective use of ground-based simulators often involves interdisciplinary collaboration between material scientists, biologists, and aerospace engineers. This necessity might seem counterintuitive. One might expect that a focus on either biological or material responses would dominate research. However, the interconnected nature of these fields underscores the complexity of simulating and interpreting microgravity systems and highlights the importance of integrating diverse expertise for successful outcomes. These insights reveal that while ground-based space environment simulation devices are crucial for space research, their operation and the interpretation of results require careful consideration of various factors that can influence biological and material responses. The report on ground-based space environment simulation devices discusses their critical role in understanding how materials and biological systems respond to space conditions. While the report presents extensive information, several important aspects that many may overlook include the nuances of different simulation technologies, the significance of collaboration with space agencies, and the implications of findings derived from these simulations. Important content overlooked. 1. Variability of simulation techniques. Many readers may not fully grasp the complexity and variability of ground-based simulators. While the report mentions various simulators such as Clinostats, random positioning machines, and magnetic levitation devices, the specific operational principles, advantages, and limitations of each are critical. For instance, different simulators create microgravity conditions through distinct mechanisms. Clinostats rotate samples to average out gravitational effects, while random positioning machines change the direction of gravity over time. Understanding these differences is crucial for selecting the appropriate simulator for specific biological or material studies. The effectiveness of each method can vary widely depending on the biological system being investigated. This variability is significant because it directly influences the validity of experimental results and their applicability to real space conditions. 2. Collaboration with space agencies. The report highlights collaborative efforts with agencies like NASA and ESA, but the significance of these partnerships is often understated. These collaborations not only provide access to advanced testing facilities but also contribute to the validation of experimental methodologies. Joint research enhances the credibility of findings and fosters the development of standards for microgravity research. This collaboration is vital for ensuring that ground-based results can be reliably extrapolated to space missions, ultimately aiding in the design of experiments and materials for actual space conditions. 3. Biological impacts and implications. The report outlines various biological responses to simulated microgravity but may not emphasize the broader implications of these findings. For example, understanding how microgravity affects cellular processes such as proliferation, gene expression, and stress responses could inform the development of countermeasures for potential health risks faced by astronauts during long-duration missions. These insights extend beyond basic science and have practical applications in ensuring the safety and well-being of crew members in future space explorations. 4. Long-term effects of microgravity simulation. The report provides a snapshot of experimental outcomes, yet the long-term effects of exposure to simulated microgravity conditions remain an important area for consideration. Research has shown that prolonged exposure to microgravity can lead to significant alterations in cellular structures and functions, which may not be immediately apparent in short-term studies. Longitudinal studies are essential to fully understand the implications of microgravity on human health and the behavior of biological systems over time. This aspect is crucial for preparing for extended missions such as those planned for Mars exploration. 5. Terminology and standardization. The report discusses the varied terminology used in microgravity research but may not stress the importance of standardization in this field. The lack of consistent terminology can lead to confusion and misinterpretations of experimental results across different studies. Establishing clear and uniform definitions for terms like simulated microgravity versus real microgravity is essential for fostering collaboration and improving communication among researchers. This standardization would enhance the reproducibility of experiments and facilitate comparisons across studies. 6. Technical challenges and limitations. The report touches on the technical challenges faced when using ground-based simulators, but it may not fully articulate how these challenges impact research outcomes. For example, factors such as vibrations, temperature fluctuations, and sample size limitations can significantly affect the quality of simulations. Acknowledging these technical limitations is crucial for interpreting results accurately and for designing future experiments that minimize these confounding factors. Significance of overlooked content. The significance of these overlooked aspects lies in their potential impact on the efficacy and applicability of research in space sciences. A nuanced understanding of simulation techniques ensures that researchers can select the most appropriate methods for their studies, ultimately leading to more reliable and relevant findings. Collaborations with established space agencies enhance the credibility and robustness of research, ensuring that it is aligned with the needs of future missions. Furthermore, the implications of biological responses to microgravity extend to astronaut health and safety. Emphasizing the importance of this research in preparing for long-duration space missions. Standardizing terminology and addressing technical challenges will facilitate clearer communication and collaboration within the scientific community, paving the way for advancements in space exploration. In conclusion, while the report provides a comprehensive overview of ground-based space environment simulation devices, it is crucial for readers to pay attention to the nuanced details of simulation techniques, the importance of collaboration, and the broader implications of research findings. Understanding these aspects will enhance the quality and applicability of research in the rapidly evolving field of space exploration. 1. Diverse simulators and their applications. Ground-based space environment simulation devices include various types of simulators, each employing distinct physical principles to replicate microgravity and other space-like conditions. The effectiveness of these simulators, such as Clinostats random positioning machines (RPMs) and rotating wall vessels (RWVs), varies based on the specific biological or material systems being studied. This diversity is crucial for conducting targeted research, as different simulators can better simulate specific aspects of the space environment, allowing for a comprehensive understanding of material degradation and biological responses. 2. Collaborative research with space agencies. Collaboration between research institutions and space agencies such as NASA and ESA enhances the development and validation of these simulators. The extensive testing of materials in simulated low Earth orbit (LEO) conditions, as noted in the provided sources, demonstrates how these partnerships can lead to significant advancements in understanding material durability in space. Such collaborations not only improve the reliability of simulation results but also facilitate the sharing of knowledge and resources, ultimately benefiting future space missions. 3. Impact on biological research. Ground-based simulators have shown promising results in studying the effects of microgravity on various organisms. From unicellular organisms like Paramecium and Euglena to complex systems such as Arabidopsis thaliana and Drosophila. The findings from these studies are essential for understanding how living systems adapt to altered gravitational conditions, with implications for long-duration space missions. The ability to replicate microgravity conditions on Earth allows researchers to investigate fundamental biological processes, paving the way for innovations in space biology and related fields. For the technical limitations and recommendations, despite their significance, ground-based simulators have inherent limitations such as the inability to fully replicate the unique conditions of real microgravity. For instance, the RPM and clinostat experiences can induce mechanical stresses that may not occur in actual spaceflight conditions. Therefore, researchers are encouraged to carefully select the appropriate simulator based on the specific biological or material system being studied. A thorough description of the operational modes and physical parameters of each simulator should be included in scientific publications to ensure transparency and reproducibility of results. 3 famous historical cases related to ground-based space environment simulation devices include: 1. The Apollo program testing. During the Apollo program, NASA conducted extensive ground tests to prepare for the unique challenges of space travel, particularly regarding the lunar environment. Facilities like the Marshall Space Flight Center and the Kennedy Space Center utilized vacuum chambers and thermal vacuum testing to simulate the extreme conditions of space. These tests were essential for assessing the behavior of spacecraft materials and systems in microgravity and harsh thermal conditions, thereby ensuring the safety and success of the Apollo missions. 2. The European Space Agency's ESA Microgravity Research Program. ESA developed a series of ground-based facilities including the Microgravity Science Glovebox (MSG) and the Random Positioning Machine (RPM) to simulate microgravity conditions for biological and physical experiments. The RPM in particular allows researchers to study the effects of simulated weightlessness on various biological organisms including plants and animal cells. This research has provided significant insights into how microgravity affects cell growth, gene expression, and physiological processes, informing future space missions and experiments. 3. The Space Shuttle Program and the National Aeronautics and Space Administration NASA ground-based facilities. NASA invested in ground-based simulators such as the Multi-Axis Trainer MAT and various Clinostats to study the effects of altered gravity on biological and physical systems. The MAT allowed astronauts to practice in a controlled environment, simulating the rotational forces experienced during launch and re-entry. Clinostats were used to simulate microgravity for biological experiments, providing vital data on how plants and microorganisms adapt to reduced gravity conditions. Research conducted through these facilities has contributed to our understanding of gravity's role in biological systems, leading to more effective experimental designs for spaceflight. These cases exemplify the critical role that ground-based space environment simulation devices have played in advancing our knowledge of space exploration and the physiological responses of living organisms to altered gravitational conditions. Future outlook: ground-based space environment simulation devices. Ground-based space environment simulation devices are pivotal in advancing our understanding of how biological systems and materials will perform in extraterrestrial environments. As we look towards the future, several key predictions can be made regarding the evolution and impact of these technologies. 1. Increased collaboration and standardization. The future will see enhanced collaboration between various space agencies, NASA, ESA, etc., and research institutions globally. This will lead to the development of standardized protocols for testing and validating ground-based simulators. Such standardization will facilitate the comparison of experimental results across different laboratories, enabling a more cohesive understanding of biological and material responses to simulated microgravity and other space conditions. 2. Technological advancements in simulation techniques. Emerging technologies such as advanced robotics, artificial intelligence, and machine learning will revolutionize the design and operation of ground-based simulators. These technologies will enable more precise control of simulation parameters, allowing for nuanced studies of how specific conditions like radiation levels or microgravity fluctuations affect biological systems at a molecular level. Additionally, innovations such as virtual reality (VR) could be integrated into simulations, providing researchers with immersive environments to better visualize and analyze experimental outcomes. 3. Expansion of research areas. While current research heavily focuses on materials and biological systems, future simulations will encompass a broader range of scientific inquiries. This includes studies on the effects of space environments on human physiology, psychological responses to prolonged isolation, and the impact of lunar or Martian dust on equipment and human health. As space exploration missions extend to Mars and beyond, understanding these factors will become increasingly vital for integrating real-time data collection and analysis. The integration of real-time data collection and analysis into ground-based simulators will allow for immediate feedback on experiments. This will facilitate rapid adjustments to experimental conditions, enhancing the robustness of the research. For instance, sensors could monitor the effects of simulated microgravity on cell cultures in real time, providing critical insights that could inform subsequent tests or lead to immediate refinements in experimental design. 5. Miniaturization and accessibility of simulators. As technology progresses, ground-based simulators are expected to become more compact and cost-effective. This miniaturization will make it feasible for smaller institutions and universities to access these technologies, democratizing space research. Consequently, a wider array of researchers will contribute to the understanding of space environments, fostering innovation and diverse scientific inquiry. 6. Enhanced focus on sustainability and eco-friendly practices. Given the increasing emphasis on sustainability, future ground-based simulators will likely incorporate eco-friendly practices. This includes using renewable energy sources to power simulators and designing experiments that minimize waste and environmental impact. The space research community will strive to align with the broader global goals of sustainability, ensuring that ground-based research does not contribute negatively to terrestrial ecosystems. 7. Human-machine interaction and ergonomics. As simulators become more advanced, the interaction between human operators and machines will be crucial. Future devices will be designed with user-friendly interfaces, making them accessible to a broader range of researchers. Training programs will also evolve, focusing on effective human-machine collaboration, thus optimizing the use of simulators in research. 8. Interdisciplinary research opportunities. The complexity of space environments necessitates an interdisciplinary approach to research. Future ground-based simulation studies will increasingly involve collaboration among fields such as biology, physics, engineering, and environmental science. This interdisciplinary approach will foster innovative solutions to the challenges of long-duration space missions, such as the development of life support systems and radiation protection. 9. Long-term health monitoring of astronauts. As missions extend to Mars and beyond, monitoring the long-term health of astronauts will become paramount. Ground-based simulators will play a crucial role in studying the effects of prolonged exposure to microgravity and other space conditions on human health. This research will inform countermeasures to mitigate health risks, ensuring the safety and well-being of astronauts during lengthy missions. 10. Preparation for extraterrestrial colonization. As humanity prepares for potential colonization of other planets, ground-based simulators will be essential in testing habitats, food production systems, and life support technologies in conditions that mimic those on Mars or the Moon. Research from these simulators will guide the design of sustainable living environments in space, contributing to the future of extraterrestrial life. Conclusion: the future of ground-based space environment simulation devices is bright, with significant advancements on the horizon. As technology improves, collaboration deepens, and research expands into new areas, these devices will play an increasingly vital role in preparing humanity for the challenges of space exploration and potential colonization of other planets. The ongoing evolution of these simulators will ensure that we not only understand how to survive in space but also thrive in the unique environments beyond Earth.